Hello and welcome to the second part of our third lecture about the scientific method. In this part of the lecture, we'll discuss a situation in which what we discussed in the previous part of the lecture, that prediction and a hypothesis are two different things, doesn't hold anymore. It's about hypothesis and predictions, about dependent and independent variables, terms that are essential in a very specific part of science, a very important part of science, hypothesis and predictions in statistics. Now, like I said before, the statistical hypothesis is different from what we discussed before. We discussed before that in general, scientific method consists of making a hypothesis, then making a prediction about the hypothesis and making an experiment to test whether or not that prediction holds. In statistics, it's a little bit different. In statistics, we make a prediction about the relation between two variables, and this prediction is actually our hypothesis, and this is exactly what we want to test. This is what we call a statistical hypothesis. Now, if we look at a statistical hypothesis, we usually have two hypotheses. The first one is the null hypothesis or the zero hypothesis, where we say that there's no association between a dependent and an independent variable. Now, we'll get to what dependent and independent variables are in the next example, but it's very important to know that you always have a second alternative hypothesis stating that there is an association between the both of the variables and that this association is actually statistically significant, not due to some random sampling error. And important to see here, and actually very logical to see here, is that both are mutually exclusive, right? If we reject our null hypotheses, our alternative hypotheses accepted, and vice versa. To give you an example and to discuss what dependent and independent variables are, we give you an example of an experiment here. Let's say you have three batches of plants and you give them three separate treatments in the way that you give one batch of plants zero point 0.1 liter of water a day, another batch half a liter of water a day, and a third batch a liter of water a day. Now in this case, the independent variable is the amount of water you give. You change the amount of water you give yourself. It's not dependent on the other variable, so it's an independent variable. Now, the dependent variable is the variable that is influenced by the independent variable. In this case, we will look at how many plants will survive depending on the treatment. So that is the dependent variable in this case. Now, there's two possibilities. Either the null hypothesis holds and we say that the number of plants that survive is not related to how much water they receive, or we have an alternative hypothesis that states that the number of plants is correlated to the amount of water they receive and that there is a negative or positive correlation or maybe even an optimum. Now, there's four possible outcomes. First possible outcome is that our null hypothesis is accepted and is also true in reality. In this case, our experiment was correct and we don't have any error. On the contrary, if our null hypothesis gets rejected and also turns out to be false in reality, we're also correct. However, when the experiment shows that our null hypothesis is true, but in reality it's actually false, we have an error. We call this a type 2 error or a beta error. If our experiment shows that we have to reject our null hypothesis, but in reality our null hypothesis actually holds, we talk about a type 1 error, an alpha error. Now, error obviously in statistical hypothesis is very important in the way we do science because it influences our outcome. It influences our knowledge generation because we accept or reject the hypothesis while we shouldn't have. Now, we can overcome that in three ways, and these three ways are always connected. Obviously, what you need to do first is make sure that your experimental design is good. You only have one independent variable, for instance. That can be difficult. Um, because sometimes you have multiple variables influencing the dependent variable. Now, you need to overcome that as much as possible because when you have that situation, multiple variables will, independent variables will affect the dependent variable, and it's very hard to separate the effect of all those independent variables and attribute what change we see is actually attributed to what change in independent variable. However, that's not always possible. So for instance, I'm in sensor research. So what we sometimes do is we make a sensor and we try to see if we can detect the target using our sensor. For instance, if you have a COVID sensor, you want to see if there's COVID in a sample present, if your sensor will respond, and if and you will also try to investigate whether or not the sensor responds more if there's more COVID particles in the sample. Now, it's not always possible to isolate the independent variables in a way that the amount of COVID particles in a sample might also be correlated with the pH of the sample. Maybe the pH changes and that influences your measurement. So sometimes it's very hard to overcome the presence of multiple variables exerting effect on your dependent variable. But you can avoid it to the best of your abilities by having a good experimental design. Two other things is you should avoid bias and you should use a control sample, and these are somehow connected also to good experimental design. Because when we talk about bias, what we often think about is conscious bias. We think about scientists not being totally objective, being biased by their opinion, by their cultural background, or by something else. Much more often, this refers to human interference, but much more often what we see is that we have unconscious bias. Bias, by the way, bias created by the way we do our experiments. For instance, sampling bias. What you usually do is when you do medical or statistical research is you can't really test a therapy or a test on an entire population, so you make a sample, a subset of people that is representative for the population. Now, in some cases, the way you make your sample is wrong, and the sample is not representative for the entire population because it's too small, or the selection of samples is wrong. There might be more old people in your sample size or more young people in your sample size, which will obviously influence the result. To avoid this, what you should do is implement random sampling to minimize the chance of bias. Each person in a population should have the same chance of being chosen, and this, if your sample size is big enough, should result in the sample being very representative in makeup for the population you want to study. Another very important part, especially for my research, is measurement bias. The data collection method needs to be chosen in such a way that the match between data and reality is maximal. If that is so, we should evaluate our technique. Measurements should be taken accurately. Environmental factors should have a minimal influence on your measurements, and multiple factors should be isolated from each other. And obviously, replication is very important to achieve statistical significance. So you have to make each measurement in triplicate or in fivefold, depending on what type of research you're doing. Again, an important example for my type of research, let's say that we use an antibody against a protein for a COVID particle, and we immobilize it on our sensor, and our hypothesis is that antibody can recognize a COVID particle in a blood sample, can catch the particle, and this will result in more negative charge on the surface of our sensor, and this is something we can measure electrically as a change in resistance at the surface. Now, how can you distinguish between multiple factors? What we usually do is we use a control sample. So we use one chip where the antibody is present and one chip where it's not. So if we see a change on one chip but not on the other, we know that it is because a COVID particle gets captured by the antibody and not just because the pH of the sample is different or the viscosity of the sample is different or we have a slightly different temperature in the room we're measuring in. So that's very important. Measurement bias, taking your measurements accurately, but also implementing a reference channel to avoid as much as possible the influence of other independent variables that you don't know about. Now, if we summarize bias, like we said, like it's very much connected to optimizing your experimental design and also by implementing control samples. So what you do is by optimizing experimental design, you can overcome bias or minimize bias as much as possible and try to overcome statistical errors. What we do in statistics in summary, is obviously we make predictions based on a hypothesis, and then we test if the data matches the prediction, which is actually the hypothesis in this case. And we check also if the data is relevant for testing our hypotheses, because this is a way of confirming that there is no error present and that our data are actually very reliable to testing our hypotheses. Thank you for your attention. I will see you in the next part of the lecture on the scientific method.